My apologies to you for stepping out for a moment. I had an urgent phone call, but which has now been made. Dr. James Prescott is our next speaker. He is received his PhD in physical chemistry from the University of Massachusetts at Amherst. He's now the director of analytical chemistry at Microchips, where he's been since 2001. He's active in the development of high-concentration peptide formulations, analytical methods development, process development, and preclinical testing. Dr. James Prescott, please. The slide changer and the
30 minutes?
20 minutes.
Thank you very much, Dr. Rothblatt, for the kind invitation to attend and talk about our work at Microchips. It's been a very full and stimulating day and 3/4 thus far, and I hope I can continue with that for you. Microchips, for those who have had the good or bad fortune to sit next to me at a meal, I probably bent their ear a little bit about what we do, but we're a medical device company. Actually, we're a technology company pursuing medical device applications around our technologies. And it's really in the MEMS, bio-MEMS space. The way we break down what we do is you could think of these as multi-reservoir arrays. They are multi-reservoir arrays. And I'm going to speak to you primarily today about our drug delivery applications, what we call active technology here. I'll define that in a moment. I'll touch briefly on our biosensing interests. Passive will have to wait for another time. The premise with the multi-reservoir arrays is you've got an implantable platform, discrete arrays, hermetically sealed. We're seeing some membranes covering these things right now, and I have a little bit of animation to make the point that on demand, we can ablate a membrane that's hermetically sealing a reservoir, expose the contents to the body, and allow some interaction with the body. In the drug delivery application, of course, then that's release of drug. It's get one dose out, you're ready for the next. Boom, there you go. You might have multi-drug arrays. In the sensing world, we're going to talk about relatively well-established sensor chemistry. Again, hermetically sealed from the body, that we can open a reservoir, expose that sensor, work it for its useful life, and move on to a next sensor. I jumped the gun there. For our passive applications, these are largely polymeric systems where we don't have quite the temporal control on exposure of those contents, as typically we're gating that time by the use of bioerodible polyesters, PLGAs, and the like. And there you go. Okay, this is, we have a cartoon here and a couple of photographs just to illustrate the concept. Some of the work I'm going to present to you was performed with a prototype MEMS device. You can see here we've got a 4x6 array of reservoirs. We're looking at the back side of it here. On the face of that, we have vapor deposited circuitry, and then it's passivated on top. And the narrow taper, I'm getting to the point where I need glasses, but I haven't gotten them yet. So I hope you can see that. If I look through a tiny pinhole, I can get my focus sometimes. But each of these dots is a thin drawn membrane over an individual reservoir, and we have a cutaway here of a single reservoir. It's not our only reservoir geometry. There are various reasons why we use this two-layer design that I could talk about later. Suffice to say, and we'll see in the next slide, that the MEMS platform is quite flexible. And so what we can do here is fill these reservoirs, and with a very brief pulse of current, 1 amp, 10, 15 microseconds, is ablate that membrane and expose the contents. Some description of the strength properties of those membranes, and I'll point out, I don't know if you can read that, but this was all published. Most of the data I'm going to present to you today has been published. Tomorrow's a travel day. If I have some collections of the papers we've done, if you're looking for some reading during your traveling, just see me afterwards. I mentioned the versatility of the MEMS platform, and so for our drug-based applications, what we're currently working with to maximize the volume of our reservoirs is rather than wet etch techniques as we have in this case, where we get inverted pyramidal reservoirs in our substrates, which lead to a very small opening through which your drug release can occur. We can maximize or play around with release kinetics by going with multi-apertures per reservoir. And also can maximize our volumes in this way. And so what you're seeing here is a single reservoir. The silicon layer is 500 microns thick. It's about a 300 nanoliter volume there. And what we can do here is, since once we ablate those membranes, our drug release is dissolution and diffusion controlled, we can come up with yet another lever for manipulating those kinetics. Okay, I'm going to speak a little bit about our drug delivery, and just before I get into any data, a couple of bullet points on why we think these reservoir arrays are advantageous. We've already heard some comments about why it might be desirable to have multi-drug therapies available for release on demand. And so what we're doing in this case is really decoupling our drug formulation from the device itself. Several times yesterday and today, people have alluded to some of the limitations of polymeric-based drug delivery systems, and specifically bioerodible polyesters as creating a locally low pH microenvironment, which might have deleterious effects on drug stability. And so we can decouple those from the drug. In this case, that's really not a relevant argument with the MEMS device, but it is for our polymeric systems. And we have some flexibility, as I just pointed out, around release kinetics. We also now have the ability to, since we can hermetically seal these reservoirs, to advance the stability of the drug form, or in a sensing application of the sensor chemistry, while it's resident in the body. That's already a challenging environment with respect to stability. Long-term stability at 37 degrees, somewhat challenging. And we'll comment in some subsequent slides on how we bias stability with our drug delivery applications. Wrong button. Okay, of course, as I mentioned, we can do multi-drug therapies. We can do exquisite control over the timing of the release with our active platform. And depending on formulation and superposition of releases, we can also approximate steady-state release kinetics. And we've already spoke about our exposure mechanisms. Okay, so I don't know when you stop calling a company a startup company, but Microchips was founded in 1999, first employees around 2000. Initially, drug delivery was our specific focus. Only more recently have we gotten into biosensing. But whenever we go out to talk to people, be it regarding prospective collaborations, looking for funding, whatever, people come back and say, any implanted device is going to elicit a peeling response, tissue encapsulation by the body, whether it's a relatively thin capsule or something greater than that. And what effect will that have on release kinetics? And so one of our earlier sort of proof of concept demonstrations around the drug delivery application was to go out and look and see if we could develop a device, release a drug, pulsatile fashion, on demand, in a chronic setting. What effect was there on release kinetics within that study? In addition, we set out to demonstrate that we could do this from a wireless, self-contained implant. And so we set out to do that, and we were successful. We have an image here of what that device looked like. You don't have a scale bar. It's about the size of a pacemaker, okay? That chip in the center is a millimeter thick, and it's 15 millimeters on a side, and that contained 100 discrete doses of leuprolide. Leuprolide is a synthetic, relatively stable peptide, small peptide, 9 amino acids. And what we demonstrated was in a 6-month study in beagle dogs that we could, in fact, perform releases on demand. What we're looking at here is average of the pulsatile releases we achieved across that time. And what we found was our area under the PK curve was invariant across the duration of that study, so conservation of bioavailability. I guess I'm getting ahead of myself. I don't have the other slides embedded here, but the maximum concentration achieved in the blood and the time to reach that maximum concentration in the blood, which would be the PK parameters of interest, if encapsulating tissue were regarding in a deleterious fashion our mass transport, those were also invariant through the duration of the study. I'll pause for a moment. The formulation of leuprolide that we used in this device was not optimized for rapid release, okay? And so we're not achieving our maximum concentration in the blood till about 3 hours out there post-release. But for this particular formulation device combination, we did not see any effects on PK as a function of healing time and residence time in the body. And in fact, if you look at the in vitro release kinetics, we do, of course, your in vitro testing is much more quantitative than you'll find in the body. We do extensive in vitro release testing of these before we put them in the animals, and the kinetics mapped beautifully in vitro and in vivo, and using a straightforward no model approach or a Wagner-Nelson model for drug absorption, our in vitro and vivo correlation of absorption was remarkable. Slopes about 0.9 there, and the correlation coefficient R squared is a little better than 0.9. We published this last year as well. And so that's our proof of concept work with leuprolide. There's a number of leuprolide products on the market, and it's not a space that we're looking to get into, but it was a proof of concept demonstration. For the last couple of years, we've been focused with our drug delivery effort on delivery of human parathyroid hormone from an implanted device. This is a combination with a device that we think has real potential for a commercial product. I think the figures here are conservative, but in the US, there's about 10 million diagnosed cases of osteoporosis. Numbers put that at about 75 million people worldwide. Implications are if you have an osteoporotic hip fracture, mortality rates a year afterwards are comparable to what you'll find with breast cancer, which is about 1 in 5. So it's a large, and I don't know if I should say unmet need, but there's a large need for better therapies to treat osteoporosis. We hear about Zometa and all the latest drugs, full-page ads in the Wall Street Journal and such, alendronate, Fosamax, and these typically are bisphosphonates. Physicians in the audience can correct me if I'm wrong, but one of the people we interact with is Bob Neer at Mass General Hospital, who was principal investigator on Lilly's phase 3 clinical study, which led to the only approved product on the market utilizing human parathyroid hormone. And the consensus we get from him is that the bisphosphonates in general are pretty good maintenance drugs at preventing further decreases in bone mineral density, but they're not particularly effective at building bone. Human parathyroid hormone is the only product on the market really that does that, okay? But there's a couple of caveats to that. Oh, and right, sorry. I've got those in my bottom bullet here. And those caveats are pulsatile administration promotes increases in bone density, favors osteoblast activity over osteoclast activity. Give this drug in a steady state fashion and you favor bone resorption, exactly what you don't want if you have osteoporosis. And so we have here an indication for which pulsatile delivery is critical. As we get involved in this space, we have a couple of parameters we'd like to achieve. Largely, the approved product, the Forteo product, is a daily injection. You would take this for 18 to 24 months before going on a bisphosphonate as a maintenance drug. And what we would like to achieve are injection-like pharmacokinetics. That's not to say that a broader pulse would not be efficacious, but we would like to achieve injection-like pharmacokinetics. And what those are, and these are taken right out of Lilly's NDA, is achieving a maximum serum blood concentration in about 30 minutes and attenuate back to baseline in about a 4-hour time period. Okay. How do we do that? How do we set out to do that? We have here a photomicrograph of one of the prototype arrays we've been working with. This is the same cutaway I showed you of the reservoir with the square sides down here. We formulate the human parathyroid hormone at concentrations on the order of, we can easily take it up to about 400 milligrams per milliliter. That's 3 orders of magnitude more concentrated than the approved product, okay? And so we don't look to these solutions that we use for dispensing to have long-term temporal stability. We simply want to use solution phase forms to dispense and fill our reservoirs. And so what we have gotten pretty good at is microscale lyophilization. And so what you're seeing in here are discrete doses of the human parathyroid hormone. The daily injection of the Forteo product provides a 20 microgram dose of parathyroid hormone. And that's what we've got in each of these reservoirs here, but that's really not a limitation for us. And depending on how you pre-treat the chip, you can sometimes get these cakes to fall out. And you can see there's some structural integrity to this. We've got these reservoir apertures in the bottom here, and you can see just like a Lego block, they've retained those features as we've removed this from the device. Okay. One of the tricks to getting one, to getting high solution phase concentrations, and 2, to getting the drug out of the device quickly, involve the formulation. The human parathyroid hormone itself, or the 34 amino acid N-terminal fragment that we're working with, does not have particularly good solubility at physiological conditions. Isotonic saline, neutral buffered isotonic saline. Solubility limits about 20 milligrams per milliliter. So as I mentioned, we can take these concentrations much higher, but we're typically doing that by creating acidic solutions to promote stability, get sufficiently below the isoelectric point, and then we can do this dispensing. Problem is, you want to get that drug back out again, you ablate those membranes, and you're exposed to physiological fluid, and are you going to get this stuff to redissolve and redissolve in a timely fashion? And so what we've managed to engineer are some acidic formulations. We'll add an excess of multivalent organic acids that are GRAS agents, free acid form. And so we can create a locally low pH microenvironment in the reservoir while these things dissolve. And so what you'll see here is, this is in vitro data, but it's, we activate a release, and within 15 minutes, we've got half the material out. And that's not a best case scenario, but with this particular formulation, what we're showing is it's highly reproducible pulsatile kinetics. And so the formulations are also quite stable. 12 weeks out at 37 degrees, we have 94% retention of the PTH monomer. 6 months, 86%. It's not a formulation that's optimized to the point of human use, but I think for the limited effort that we've put into this, it's a demonstration that this is, in fact, achievable. Okay, and so where we've gone from this is 2 in vivo studies, preclinical studies. I was unable to include some of my PK studies in this presentation, but let me comment. We wrapped up a preclinical PK study in a beagle dog model about 2 weeks ago. Devices were resident for 3 months in those animals. It's a sufficient time for healing response to occur and for the most part to mature. And we do get our pulsatile release kinetics and find those conserved throughout that duration of time. We're back to baseline within the 4 hours that we set out to achieve. So we're quite happy with those results. And our next step will be occurring in the 3rd quarter of this year, and that's a efficacy study using an overectomized rat model for osteoporosis. That will be our first efficacy study around our drug delivery applications. And so watch for that one. Okay, and then what I've got here are a couple of models, device concepts. Device concepts are a funny thing because anybody versed in the programs can make these things, but we think that these are realistic. We've tried to put some realistic boundary conditions around these, and think that these are achievable device sizes for this application. I think we just saw a couple presentations ago about how we can get the integrated circuitry built into chips themselves, and that realizing that in a device like this would go a long way towards miniaturization of these devices. Okay. In just a couple of slides also, I'd like to make a brief comment on our continuous glucose monitoring system. This is the main thrust of the company at present. Okay. The concept here is that a lot of your glucose sensing chemistry, enzymatic chemistry, is relatively old chemistry, relatively well-established chemistry, and there's been no shortage of activity trying to develop more robust sensors around the old chemistry. And what we are finding we can do with the multi-reservoir arrays is take an array of discrete sensors. This is an old cartoon, but you can see inside an interdigitated electrode, okay, and it's hermetically sealed again until we ablate that membrane, expose it to the body, and you've got direct contact with interstitial fluid. I'll show a figure at the end showing how a current generated redox chemistry, redox of glucose, electron transfer, small current. You can measure a current correlated with glucose concentration. And rather than trying to develop more robust sensors that will survive long periods of time in dwelling, you can have an array of sensors, each with perhaps a finite life of weeks to a month. And if one fouls or loses activity, you can open the next reservoir. Okay, and so our advantages here, we think, are that there are bullets here, and you guys can read them as well as I can, but the current approach, as I'm just mentioning, has been to try and develop thicker layers to last longer, and so what we're finding is we can use, simply by sealing these and opening them on demand and letting them go for a finite life, we can still get a long sensor life by having an array of these sensors. The semi-permeable membranes they often use to protect these indwelling systems are tend to fouling. Again, we can open fresh sensors and get away from those problems. Okay, no sacrifice for longevity. Here we have a cutaway of the device. I'm not going to comment too much about this except to say we're looking towards our first human trials with these devices in late '09. And as I promised earlier just a demonstration of the concept in vitro. These are some sensors that we have running in the lab right now. And what you're looking at is how the current steps with changes in the glucose concentration in our in vitro test system, and beautiful linear response curve over a range of glucose concentrations that are of interest for this type of application. Okay, and so I'll leave you with these messages, and thank you all again for your time and patience.